All right, it's 1 minute past 1 o'clock Eastern. We'll kick things off. This is the Sona Nanotech webinar for investors. My name is David Regan. I'm Chief Executive Officer of Sona Nanotech, and I could not be more proud to be here today with my colleagues Len Pagliaro, PhD, who is our Chief Scientific Officer, and our scientific advisor and the principal investigator on our current efficacy study, Dr. Carmen Giacomo-Antonio. And I say proud because we've got some results coming out of this study that are really interesting and frankly bear delving into the details to understand some of the nuances. And unfortunately, we have Dr. Giacomo-Antonio here to help us with that. So we will make haste to move into that data quickly, but I do want to set some context first. And of course, as an overview for folks, Sona is developing a therapy called targeted hyperthermia therapy, which is the intratumoral administration of gold nanorods into solid tumors and then the subsequent exposure of those tumors to near-infrared light, which gently heats the tumor from the inside out, killing cancer cells selectively. That said, with the study data we're showing today, we're starting to see something even more interesting. Of course, as a public company, we will make a number of forward-looking statements that we need to point you towards our disclosure documents for and the risks of future unknowns associated with that. So Sona Nanotech makes gold nanorods, and we make gold nanorods because gold nanorods are highly functional amongst all the nanoparticles out there. They have an aspect ratio to them, allows one to modify both the length and the width, which then can resonate with different wavelengths of light device. They are uniquely biocompatible. No one else makes gold nanorods without the use of a toxin. We have that. It's patented in one territory, pending in further territories. And perhaps most importantly, these materials have been validated by an organization in the United States called the Nanotechnology Characterization Laboratory that has assessed 7 batches now of our materials and found them to be free from endotoxins and any microbial contamination, which are the number 1 or 2 issues. Questions already starting to come in. I should point out also, yes, there is a chat box at the top of your page. Feel free to press that chat box and add questions, and we'll get to those towards the end of the presentation. In any event, the NCL was set up by the FDA and the National Cancer Institute to accelerate the progress of nanomedical applications such as ours for smaller developers by giving them access to very sophisticated equipment and personnel. And we've been really fortunate to be working with those folks. This is a program that Len and his team were accepted into several years ago, a very prestigious program whereby they're very selective, and the therapy developers they allow in, and we've benefited tremendously from that, the work we've done together. So now, this is the context part. Immunology has been the significant driving force in oncology for the last 10, 15 years. 2013, it was, I think, named the Science Development of the Year. And in that time, sales of the drugs associated with immunology, of course, immunology is the practice of helping your body to fight cancer on its own, using its own devices. Of course, the issue being that your body thinks the cancer is yourself and won't attack it. Now, these drugs in 2021, 31 billion sold. That number in 2003, I believe, is up to $50 billion US sold. A lot of money being put into these drugs across the board, a lot of the major makers. The first one is, in fact, the brand name is Keytruda from Merck, is the #1 drug, and it's the #1 drug in the world across all domains. And there's a reason why. Because, and here's some data from Merck, in fact, showing how these immunology drugs, and this one in particular, Keytruda, combined with chemo, improve survival rate in this one study by 81%, 5-year survival study. These drugs, when they work really well. What they do is a big, big advancement, and that's why it's been the full push. However, the issue is response rates are still very limited to this therapy, depending on the type of cancer. So on average in this study, 21% response rate. So in other words, you've got a 1 in 5 chance that this therapy that works so well will work for you or for the particular tumor in your body. That begs the question, why doesn't it work more frequently? And there are a number of different reasons behind that, the research shows. However, the number one issue we believe is the weak tumor antigen or loss of tumor antigen expression. Now, what does that mean? At this point, I want to introduce our principal investigator, Dr. Carmen Giacomantonio. We're very fortunate to have as a scientific advisor to the company too. Carmen, you want to tell us a little bit more about this?
Thanks, David. So this is exactly as David was saying. So immunotherapy has changed the world. We know that. We hear it in every disease, in every, not just in cancer, but in all kinds of other benign chronic diseases. But we're focused on cancer because there's what we've discovered is that we can actually unleash the immune system in a more elegant way, in a more specific way, in what people would call precision medicine. So what we're capitalizing on is this. The current immunotherapy models are based on checkpoint inhibition. That means this. Your immune system engages a tumor that you grow. As the tumor grows, the immune system responds and creates an army of cells that would attack it. But because there are mixed signals, or because there's a quieting of the attack, or because the immune system runs out of energy, so to speak, these things shut down. So what investigators have done is they've said, well, there are many checkpoints. Let's or disinhibit more of the checkpoints, and we'll recreate the immunity. And sure, it does. It causes the immune reaction to happen, pushes the equation from left to right. But the consequence is that your immune system, with all the checkpoint inhibition, begins to be disinhibited against oneself. And so autoimmunity is the toxic and at times lethal consequence of too much inhibition. So what Sona has developed and done an excellent job is creating a toxin-free nanoparticle that we can inject into a tumor. And there's many ways to deliver gold. Some people inject it intravenously. We've chosen to directly inject into a tumor because it limits the toxicity. It limits the systemic exposure, and it really gets us to the cold face, if you will, to where the immunity is happening. Inside the tumor, the gold nanoparticles, once deployed, can be activated. And again, as David said, passing a near infrared light through a gold nanoparticle of a certain shape and size will cause it to vibrate. And the vibrational energy creates heat, thermal energy. And that thermal energy is what we capitalize on. And the principle is this. The thermal energy, if it gets to a point somewhere between 42 and 48 degrees, the sweet spot being 45-ish, that the tumors will go, cancers, although we think about them as animals and beasts and being robust, they're actually quite frail. And stressed at 45 degrees, they'll go into a death cycle called an apoptotic death. Now, the beauty of that is when a cell dies an apoptotic death, it doesn't denature the protein. If we took a blowtorch, if we cauterized the tumor, if we annihilated it, we would destroy the protein such that that protein wouldn't be of any value to the immune system. But when we do it this way with the gold nanoparticles, we kill the cell, and when a cell is killed, your immune system comes in to clean it up. And as it cleans it up, it packages all that protein, and in the course of being an immune system, it does the only thing it knows how to do, and that's it shows it to the rest of the immune system. And if the protein that it shows is interesting, if it's foreign, as much cancer protein is foreign to your body, then the body will attack that based on that protein signal. And that is new immunity called innate immunity. And so what we've done is we've taken that principle, and then we've added the notion that if that's an immune interaction, we have immune agents in the clinics we use today. I use them all the time, interleukin-2 being one of them, a very common, very Canadian, by the way, strategy, but being adopted around the world now. But we were actually leaders in this, in terms of the intralesional, intratumoral delivery of interleukin-2, which is also called T cell activating factor. It makes the T cells aggressive. And so if the T cells are aggressive and they have a new antigen to see, the immune response should be better. And so this was the fundamental basis of our experiments, and we're going to show you some of that now. So we went to the lab. We used this notion, if you think about it, is what I just said was all cancers create abnormal protein. So we're showing abnormal protein in all cancers. So it's tumor agnostic, if you will. Doesn't matter what the diagnosis is. So we look to the animal models. Next slide. And so when we look, we have three objectives in the research. The first is that we're going to shrink the tumors, and we know that the gold will heat up the tumor with the activation through near-infrared light, and the tumor will die. And of course, that's a physical shrinkage. And so the question should be, well, what does that mean? Because, as Donald Trump said, we can inject Javix into a tumor and kill it. That doesn't prove anything. So then the next thing is what happened when we did that? We may have exposed antigens, so we believe we're enhancing that immune response. The real test of immunity is if it's an immune response in the tumor, is it robust enough response that it would affect potentially other tumors elsewhere in the body? And that's this abscopal effect that we're all looking for. It's the unicorn in the room, if you will. Next slide. So we did the experiment. We took in this model, this is a triple negative breast cancer. It's a 4T1 for the scientist minded in the room, but it's a 4T1. It's a standard industry standard model of immunity. And most of the immune research is actually done in mice, and mice aren't people, so there's no question. But the cell-cell interactions are very translatable. And as you know, we're a highly conserved universe. So we took 4T1 triple negative breast cancers, and the experiment goes like this. We drew two tumors, one in the left flank, one on the right flank. The left flank is the treated tumor. The right flank is the metastasis, if you will, the other tumor. And what you're seeing right there is the ta-da. That's the ta-da slide. When you look at the agents that we used interleukin-2. That's my standard therapy. We used a control. So this is just phosphate buffered saline, just water. This is the heated gold nanoparticle, and this is the combination. And so the exciting thing in this, if you see nothing else right from the start, is, wow, this line is flat. And this line is the effect of the combination of the hyperthermic therapy and the interleukin-2. So this 1-2 combination is better than gold nanoparticle therapy all by itself or interleukin-2 by itself. Now, interleukin-2 has an effect. The line starts to flatten. Gold nanoparticle, actually, this is one treatment with near-infrared light, and when you see you check the tumors the next day, 80% of them are shrunk. But what happens over time is there's a regrowth. So next slide.
And Carmen, while you're doing that, speak to the idea as to because we had an excellent question around, what were our expectations going into this?
Oh, so that's a great question. So what I expected to see was divergence in the line. What I expected to see was the gold nanoparticle would shrink the tumor, and the interleukin-2 would shrink the tumor, and both of them would shrink better than either one individually. What I never expected to see was this line flatten. And this is the punchline. This is statistically significant across the board. These error bars these little whisker things don't overlap. And that means that is clearly different than either one of them. And so this is not only additive, it's synergistic. It is exactly what we had hoped to see. One gold injection, one treatment with the laser, and then a series of interleukin-2 injections, which are essentially fueling the immune response to continue with the drive from left to right. Because, and I'm going to show you a slide in a second to explain why, the immune system is naturally designed to check itself. And so if you can do something as simple as push it to the right, then that's what you do with the interleukin-2, series of injections. And the next slide is actually, it's just as intriguing so what I said earlier was, in the tumor that we're treating, that's on the left side of the mouse, we're seeing a tremendous response. On the right side of the mouse, where we haven't done anything, we haven't given anything systemically, we haven't given anything into the tumor directly, this is all the effect of what's happening on the opposite side of the animal. And we are seeing a tumor response along here that's not completely divergent, but it is a response that is indicative of a systemic response. This is systemic immunity, and this is the abscopal response that we hope to see.
And Cameron, can we just point out and remind folks that on the left-hand axis here, this is the tumor volume. The blue line is the control. So left to its own devices, the implanted tumor just grows and grows and grows. And of course, this is days down below. So in this untreated tumor, to see the therapy that was given to another tumor in the animal, creating a reduction in the size of its volume is a significant result.
Absolutely. Two things of significance on this slide. You can pick at the slide all day, but here's the other thing. You see this is the blue line, the control line is going up essentially in a linear equation. So is the IL-2. The interleukin-2 given into the other side of the mouse did not have a systemic effect in and of itself. It wasn't
and that's effectively standard of care today. Is that fair to say?
That's exactly right. And so that's what we see in the clinic. When I inject tumors with IL-2, we don't see abscopal responses from IL-2. We don't see the tumors beyond the field that I'm injecting respond. What we see is a local response. So the fact that there's a response in another tumor means we must have generated new immunity. Now, I'm going to show you some of the immune data. We're just going to go by this just so I can try to convince you. Next slide. That there is immunity here. So these are immune cells. CD4, CD8, these are CD is cluster of differentiation, and these numbers are molecules that we measure to tell us what we're looking at. There's
Carmen, can I interrupt for a second? Yeah, can you provide a little bit of context as to this for the lay folks amongst us here and amongst investors?
So what we've done is we've taken the animal, and then we've taken the tumors, and we've taken the infiltrating cells, and we've looked at the response both in the tumor and systemically. And so in the tumor, when we look, we see the cells that are invaded. So we ostensibly drove those tumor cells into a death cycle, and then there was an infiltration of cells, and these cells are all immune cells. We stain them. This is called FACS analysis. We take it out, we homogenize it, and we put it on a kind of a scanner, and we separate the cells based on their features, and that's the molecules that they express. So what we're seeing here is these CD8 cells are the cytotoxic cells. They're the cells that are actually killing tumor cells. And we're seeing that if you look over here, the control versus the gold nanoparticle plus IL-2, this highlight tells me that that's statistically significantly different.
Which one do you want to? Which one did you?
The far right. Yeah, the upper far. So that's
CD8 and PD-1.
Yeah, and it's also expressing PD-1. So not to confuse the talk, but that PD-1 is important because that means that's a checkpoint. So these CD8 T cells are doing what CD8 T cells do. They're starting to turn on the checkpoint to say, should I continue to fight? So I can do one of two things at this point. I can give more interleukin-2 and override the PD-1 message, or I can give a checkpoint inhibitor. Now, if I was in a clinic and I had a PD-1 inhibitor like Keytruda, I'd say, we'd want to pair this with Keytruda and get a bigger response. And so this in and of itself tells us there's an opportunity here to have a combination therapy that would be more effective, again, than either one alone. And IL-2 works, PD-1 inhibitors will work, other checkpoint inhibitors will work too. Down on the bottom side, what you're seeing is that you're seeing memory. So on the second from the left on the bottom, you're looking at CD8 positive CM. That memory cell means that we're generating an immune cell that has an ability to find disease somewhere else, i.e. that other tumor, or somewhere else in the body. Any new antigen, those tumor cells are going to circulate, and when they encounter the antigen that interested them in the first place, they will be reinvigorated to fight. And so this is the immune data that we needed to convince ourselves and hopefully everyone that this is an immune response, not just a hyperthermic ablative phenomena. Next slide. And the last slide I'm going to show you in terms of the immunity is the lymph node. So when you're looking at the tumor, that's what's infiltrating. These are the cells coming and attacking the cancer. And this is the work that's happening in the lymph node, just like in us. When we have a sore throat, the lymph nodes in our neck are actually the ones that are doing the fighting, sending out the fighter cells. And on your left on that side, you're seeing the FOXP3 means that there are T cell, these T cells are kind of like regulatory cells. So you want to be aware that you've got them, but it also tells us that the immune system is regulating itself, which is important. So, and then again on the right, you see to the far right, these are natural killer cells. And natural killer cells are the kind of the primary cell of the innate response, the first response we get. The natural killer cells are activated, and we're seeing there's a statistically significant activation of natural killer cells relative to those controls with our gold nanoparticle IL-2 combination. Next slide. So so we've got a two-step situation here. We are heating the tumor. When we heat the tumor cells are the ones that are fragile, and they go into the death cycle. The rest of our cells don't. We've all been in, well, maybe we haven't, but we could tolerate 45 degrees of temperature. We won't go into an apoptotic death. And in fact, it's more complicated than that. Obviously, this is an exposure over time. There's real science and medicine happening here. But the other thing is, so this is a patient. This is a melanoma patient, and this is the kind of the Super Bowl of challenges. This poor gentleman, a patient of mine, had a melanoma just below his ear, and it started to spread. And those are the draining lymph nodes. Just like if you got a sore throat or you've got an infection on your cheek, this is the lymph nodes, and this is the skin that will respond to that. Well, the melanoma distributes in that tissue, and then it grows up, and it overwhelms. It's just not seen for a number of reasons. We think in this case, given that he failed all checkpoint inhibition, that the antigen just wasn't sufficient. On the left-hand side, what you're looking at is this is a blood cell to give you a sense of scale. That's a red cell, and those little specks are the nanoparticles. They're truly nano. They're nano in size. That's a whole cluster of minuscule particles that distribute throughout the tumor. And then in the middle, you see the cartoon of the near infrared light being passed through the tumor. It penetrates about 2 centimeters or so into tissue, and everything that it penetrates through that has gold nanoparticle will be heated in the concept of that. And that heat will cause the apoptotic cell death. I'm stressing that word apoptosis because it's a programmed cell death that results in sparing of the protein that we can take, or the immune system can take advantage of. And then the other thing is that these new antigens, neoantigen, an antigen is just a protein that's interesting to an immune cell. And so these new protein antigens awaken this innate response. That's the initial response to anything. And so what you've created is a personalized vaccine, if you will. We can talk about whether it's not a vaccine or it is a vaccine, but it is the concept and the premise of what vaccines are. So what would we use this for? We started out in a triple negative breast cancer model. But that's an industry standard. We are now doing our melanoma models. If you look at where's the likely place for us to find a purpose in the clinics, it's the kind of patient I just showed you. There's a lot of stuff happening in breast cancer. There's a lot of work happening in immunotherapy in breast cancer. So that's on the runway. But what's an immediate need for are these patients. These are the advanced stage 3 with this in-transit disease, or the patients with stage 4, meaning they've got disease throughout their body, but also involving the skin. That's what M1 is. And they failed immunotherapy. They failed surgery, or they've had toxicity to one or the other, or surgery is just prohibitive. And so or they've had a partial response. We see this. Some tumors respond to immunotherapy, some don't. We can try to generate new immunity using this process. So that's our target audience. And so the next slide.
Carmen, I'm just going to ask you this. So we're focusing on, and this is a bit of a pivot for us, as we've talked about colorectal cancer in the past, but we think for a number of reasons that melanoma is the place to go first, and we'll talk about that later. But this is later stage 3 and 4. Why not earlier stages?
Well, because that's a very good question, because in medicine, there's just tradition is we start with the worst and we move back to the best. There are lots of options in the earlier stages, cut it out. And if you can cut it out, that's what you're going to do first. So it's going to have to be the late stage that's come back. And so stage 3 means it's in the nodes, in the lymph nodes. Now, you can give me an argument where the nodes are too bulky and I can't operate on them. I'd say that's a place to do this too. But it's going to be something that we're going to have to prove the efficacy and the safety in a patient where there was truly no other option, so that we can justify learning what we don't know, if you know what I mean. So the intended use is for these people. What we want to do is create the hyperthermia. We want to show that we can create new immunity in things that we can see. but we can't resect either because the surgery is more morbid or the extent of the disease is beyond resection without some intolerable toxicity.
Right, want to move on?
Yeah. So this is the algorithm. When a patient comes to clinic, we follow this algorithm of care. If they're resectable, we resect. If we can give a local therapy or a systemic therapy, we give it. If we have to dissect nodes, we dissect. If it's unresectable or it's recurred time and time again, then we're left with, what do we do? So this often if it happens in a leg or an arm, we can do an isolated limb perfusion. If it happens in something, we can inject. IL-2 is what I use. Imiquimod I use in Canada. TVEC is the United States, kind of like an IL-2 thing. And then we're down to some kind of systemic treatment. And the systemic treatments, if they work, that's great. They often fail in the skin, and so if they fail, we have to come to what's next. So then we go to the guidelines, and we use the NCCN, the National Cancer Collaborative Comprehensive Cancer Network guidelines. And if you look in this guideline, it says workup. So we've done the workup, and now we are at the stage where we've got that area in the square to the left with the circle around it. Now, we've biopsied it, it's melanoma, we've tried everything, it's still there. Now we go to the middle column, which is your initial treatment. And that middle column is very extensive. What it means is there isn't a number one choice. It's use what you have available for you in your system to the best of your ability. That column is begging for a solution. That column should be the line A, choice B, but it's not. It is all of those things are completely acceptable because there's nothing that actually works very well.
So it'd be fair to say these are the alternatives to what we're proposing.
Yeah, that's exactly right. And that long list tells you none of them work very well individually.
Right.
What we're proposing is something that should put this to the top of that list.
Excellent. Thank you. Now, Lynn, do you want to talk to us a little bit? Tell us about where we've come from, what's been done before, how far out there are we?
Sure. So although this is in general a new kind of technology moving toward the clinic, there are many aspects of it that are known and have been proven that out there. It's well known, as Carmen mentioned, that heat can be used to kill cancer cells. And the reason is the cancer cells are more heat sensitive than non-cancer cells. So there's a degree of selectivity built in there. There are photothermal treatments out there, none yet to the market that we know about, but in development in clinical studies. We think we have advantages over those photothermal treatments, both in terms of the quality of the nanoparticles and also in terms of the quality of the light device. And so that's an important differentiating factor there. The FDA has approved nanoparticles for injection into humans in clinical studies in several different settings, at least one of which is photothermal, and then another of which is a thermal therapy that is not photothermal, uses magnetic energy. So that there are precedents for that. And then finally, there's a lot of literature, including our own, around the efficacy of THT in small animal models and we've also done this in comparison to the standard of care, which, for example, could be a drug. And you look at the efficacy of the THT, the drug alone, and then the combination. And the combination is extremely effective. And in many cases, the THT alone is more effective than the drug itself. And so there's a lot of work out there, a lot of precedent out there supporting what we're doing. Now, in terms of evaluating next steps and looking at the market in terms of what we're doing, of course, we have to deal with physicians and patients, but also with providers and payers, and we've got to look at the market scenario. We've done a couple of important things. First of all, we've engaged a panel called Excite in Canada, which is an excellent panel put together with practicing physicians, surgical oncologists, as well as representatives from payer organizations. And they've vetted what we're doing extensively. We've had a number of meetings with them and very valuable feedback from them with important advice on positioning our technology the right way. And in fact, their feedback is one of the things that alerted us to the potential of using skin cancer, melanoma, as a good first in human indication. Could we go back to colorectal in the future? Of course, that's possible. But as first in human, melanoma is an excellent choice. Then the second thing we've done is we've already started working with the FDA. We've had our first pre-submission meeting with the FDA. Now, when you go through a regulatory process, you have to submit a package. In our case, it's called an IDE, an investigational device exemption. And before you submit that package, you have what's called a pre-IDE meeting. We've had our first one now, and very valuable feedback there from the FDA. A large panel of people from the FDA responded to a number of our questions to them, and we now have minutes recorded from that meeting. And so good feedback on moving forward.
Thanks, Len. And the one thing I would point out here with both of these entities, the reason we put this in here is that our development plan has effectively been developed with feedback and input from both of these entities. And Len pointed out that with payers are a significant constituency. We were fortunate to have on our panel there are 4 payer organizations. One of those individuals who represented one of the largest payers in the US has asked to join us at our next pre-IDE submission meeting with the FDA sitting on our side of the table. It's really valuable, as we, I think, perhaps mentioned previously, but the folks from the NCL attended with us, had offered proactively based after they've analyzed our materials and sat with us on the other side of the table in our first meeting. This is why we're really feeling strong, and this is why you're seeing some shifts and pivots as we focus in, and even as Carmen talked about the specific focus being late stage, stage 3 and stage 4 for melanoma. It's been due to the input of these folks, which we're extremely fortunate to have. So where does this take us? We're moving quickly. We've got a whole host of partners that have been vetted, a lot of time spent on finding the right partners and understanding what it is we need from them in order to take that forward. So that is done. The panel from Excite, we've had 3 sessions with them. These are senior physicians from name brand universities, cancer centers. They are at the top of the field and will become advocates for us as we move forward, we believe. Had that first session with the FDA. Len has mentioned we'll be having another. Gold nanorod GMP. Gold is a good manufacturing practice. It's required to go into the first in human studies. Needs to be done in an outsourced facility with a top tier global producer who we have vetted and selected. Efficacy studies, of course, are pending and working with Dr. Giacomantonio and his study group. And those we're in melanoma. We're in the triple negative breast cancer. By the way, triple negative is the worst type of breast cancer, so really stress testing it in that realm. We move on from there into a melanoma model and a colorectal cancer model. So lots of work being done in the Giacomantonio lab, and lots more data to come from that we hope to replicate what we've seen today. Next step is initiate the safety, feasibility, and biocompatibility studies that are required for that IDE submission Len mentioned. And of course, we need to initiate the manufacture of gold nanorods in the GMP facility. That's pending because, as we say, we've selected the partner. All this leading to the ultimate goal to get into humans, or frankly, if I can paraphrase Dr. Giacomantonio, what he really wants is to get into clinic. And so that this can help people, because we have a strong belief that will be the case. To prove that out, we need to get into a first in human study, and we're moving as efficaciously as possible and efficiently as possible to do the fastest path we can to get there in order to prove this out and warrant and get approved the right to go into a pivotal first in human study. We say some folks will have seen this slide. We've engineered the right gene pool here There's been a transition over a few years, and we're very fortunate to have a board that hits well above its weight and quite appropriate. When Carmen starts talking about this procedure, this THT as a vaccine, we have Mark Levonin, who led Sanofi Pasteur for many years. Dr. Walter Straps, who's CEO of a biotech in Boston. Neil Fraser, longtime president of Medtronic Canada, leading device manufacturer, and Jim McGann, a venture capitalist and capital markets professional with Numis Financial. We have our management team across the board, and we're also very fortunate to have advisors, folks who will pick up the phone when we call with questions across the board, including on the medical side, on the science side, and on the product development side. So many facets of this covered. So with that, we're really very excited as to where this might go, and we've got lots more data to come. Before we get into the questions, and again, I'd encourage you, if you wish to ask a question, just press the chat box at the top of your screen. But Carmen, if I can kick it back to you for a second and ask, from the perspective of the study, what are we expecting next to come?
So there's a couple of things within this study, we're repeating it. Of course, we have to, and we've got the early data that says it looks exactly the same as we've done before. We're modifying some things to see, varying the range and size of the tumors, because tumors don't come in the uniform shapes or sizes. And then the next study is going to be the melanoma study. The triple negative breast cancer study is very exciting, because we think that that's a clinical entity that we will be able to move into fairly quickly once we've got some comfort in the in-human situation in the melanoma. And so we will have that data coming out. The melanoma, for anybody that's done research with the most melanoma model, it's not a highly immunogenic model, so we're even more excited to see that we can generate immunity in a fairly cold tumor. The concept of hot and cold being hard to get immunity to be interested in, hot being the ones that all the immune system see. And then we've got the colorectal cancer model, which is coming out not third, but kind of parallel to the melanoma model. And in the colorectal model, we're actually looking at a vaccine protocol, which is super complicated. So that will follow on the heels of this. But these things are underway. They're kind of overlapped, as not done in parallel and not done sequentially. They're overlapping, and as we learn from one, we modify another, so that it makes us more nimble and try to not ask as many unnecessary questions. Right. So that's where we are. Yeah.
Excellent. So let's kick it over to some questions here. There's a great question here asking about the idea that you're focusing in on the distant tumor. So treating one tumor and having it have an effect on a second tumor, having that hope that it'll have an effect on the second tumor. Can't this just be, wouldn't if a dose of chemo effectively hit all tumors in the body?
Great, perfect question. So yes, for the chemosensitive tumors, the melanoma is essentially chemo-resistant. There isn't a chemotherapeutic agent that's been discovered yet that has any kind of reliable response rate above 10%. It's so rare to use chemo now. It's people have to be reminded that it's an option when all else has failed. So the abscopal effect that we're talking about is that we actually see it in melanoma now occasionally. I use interleukin-2, intralesional. So when you inject a lesion, it's just the only reason to inject it is, well, there's two reasons. One is you can get to it, and the second is you reduce toxicity. All of the toxicity that we see in chemotherapies and in lots of other systemic therapies are the unwanted effects of activating systems or irritating systems that we don't want to irritate. And so forever, since I was a medical student, we've had this notion of a silver bullet. The intratumoral injection is the silver bullet, and all of the immunity happens within that tumor and the related draining lymph node. And so what's elegant about this is that we are simply limiting the attack to the one that we can access. And we are right in showing that we're creating new antigens, so other tumors disappear. When I inject a tumor in the clinic with BCG, which is an immunizing agent for tuberculosis. It treats the tumor by creating a big ulcer, but that ulcer causes tumors to die through this process of apoptosis, and the antigen get presented, and every now and then, actually 30% of the time, other tumors die, and we've published that. It's a messy process, and in using a tuberculosis vaccine carries its own problems in the case of context of immunity. So this gold nanoparticle is far more elegant in that context.
And there's an associated question here. Are there any known limitations in administering interleukin-2? Accumulation in the body, for example?
No, so it doesn't accumulate. The half-life is literally seconds, but there are limitations. When I started giving, I don't want to say how long ago, but it's almost 30 years ago, I started giving systemic interleukin-2, and it's super toxic. It causes what's called leaky vessel syndrome. And it's the same thing if you can imagine having COVID, a catastrophic infection where your immune system overwhelms your body, you decompensate, you become hypotensive, and you end up, and these patients have to go into intensive care units. Interleukin-2 was discovered in 1975, first used as an experimental tool, brought into clinics in 1985, and used with some interest until about early 90s, when too many people got sick from the toxicity. But it had a complete response rate of about 12%. The response rate was low, but that's low relatively speaking, but everybody responded, which told us it was an immune response. So, but its limitation is the amount you can give in one dose. But I can put, relatively speaking, thousands of times the volume of interleukin-2 directly into a tumor than I can giving it intravenously and trying to distribute it beyond that into the tumor.
Okay. So there's another question here related to the idea that our targeted hyperthermia therapy will, deliver this 1-2 punch. In other words, the THT softens up, if you will, in my language, the tumor, opening it up, exposing those new antigens, allowing the immunology drugs to come in and do their job. But we only, in these studies, administered one treatment of THT. Would we recommend doing a second treatment? If so, when?
It's another great question. So the answer is perhaps. When is TBD. So probably within the first 48 to 72 hours, when we've got this apoptotic, we saw based on our first experiment that 24 hours after activating the gold nanoparticle, 80% of the tumor was gone. So 80% of the tumor does it mean that we didn't wait long enough, or we didn't create an apoptotic death in the other 20%? We don't know. We think it's because we didn't create an apoptotic death in the other 20%. As you saw without the IL-2, the tumor started to regrow. So it could mean that we just have to add another exposure of near infrared light, because the gold distributes throughout the tumor when you inject it. And it could mean you need more gold. This is part of the design. Our next set of experiments with the big tumors, we're looking at both of those. And so I'd be able to better answer your question 4 weeks from now. But not either way. We're giving gold therapy has been used in people for years. We used to treat rheumatoid arthritis with gold, big systemic doses. We're giving minuscule amounts relative to those. So we've gone a long way already to address toxicity concerns by simply pivoting and just injecting the tumor of interest, knowing that we can create a systemic response with the local treatment, i.e. vaccine.
Yeah. And Len, can you comment on the discussions we had with the FDA and their thoughts to us, as shared to us, on systemic versus intratumoral, and what the implications might be for the studies required of us?
Sure, yeah, and I see there's a question on that as well. So a couple of big things here. If you inject pretty much anything systemically in the eyes of the FDA, the risk goes up, as opposed to a local intratumoral injection. And so that was one of the issues that made melanoma as an initial target more attractive. If we go with that and we do intratumoral injection, then the safety risk in the eyes of the FDA is reduced significantly. And what that means is that we anticipate, based on the feedback we've had so far, that we will need to do studies only in rats. In rodents, we have mouse data already, and we'll have to get rat data for safety, but we will not need a large animal model in terms of a safety model. So that's a really good thing. It's also worth pointing out that the safety profile of the nanorods is extremely good. from data in mouse models where we've put basically as much as we can into mice, and there don't seem to be any harmful side effects. But again, the FDA, as you know, is very risk averse, so we have to take these things one step at a time with humans.
And a related question here that ties right into this, which is, can we take advantage of any potential treatment of last resort, which I think refers to compassionate use designation? Carmen, do you want to speak to that ability?
Yeah, and that's another excellent question. And the answer, the short answer is yes. You can, but if you from a clinical strategy, I'm speaking like a zealot now. I know this is going to work. I'm very excited to get this in the clinic. If you start with just compassionate use, you've absolutely limited yourself to the rarest of the rare. And so you've created an additional layer of work that was probably unnecessary. And there's a bit of we've done a lot of things under compassionate use, under the auspices of compassionate use that make it sometimes let us do more questionable things. So yes, we could definitely. I would rather see us take this into melanoma in a justified, approved way, and try compassionate use in a more challenging tumor that we haven't treated with it before, for the reasons that it's already been approved and vetted in a disease. So now we're really just focused on the biology of the other disease, not the device itself.
Right, and there's certainly, in discussions with Health Canada, there's certainly the feedback that a certain level of safety and biocompatibility data is required before allowing something like this. So in any event, we'll need to proceed with some of the studies we have lined up here. Another great question here. It was previously discussed that THT was viewed as a two medical device system, which it is. There's the gold nanorods, and then there's the light device that is applied towards the tumor. Two devices. Does the addition of immunotherapy change this, or is the immunotherapy viewed as a bolt-on to the treatment?
Well, I take a crack at that. So in our case, the immunotherapy, the stimulation of the immune system is coming from the heat that the targeted hyperthermia generates. Now, if it is used as an adjunct with a treatment such as IL-2 that a physician such as Dr. Giacomo is administering, then the THT component as an adjunctive therapy does not have to be vetted as a drug. It is a device. It's a two-device system that's being used together with a drug to treat the patient.
Excellent. A few more great questions here. Is there a negative significance of so few mice in the study? What is the significance of the first chart, the green line after 10 days goes back up? We didn't mention that. That's a good point. That's a good catch, actually.
Yeah.
Carmen?
So what it means is, and we kept the slide, and I don't know if we have it in this deck, David. But
yes, there's the back there.
Yeah, let me go back. Yeah.
So that's a very good and scientific question. So the uptick could be that there was a small amount of tumor in one or more mice that started to regrow that would need another treatment with IL-2. That's one thing. But we do know from that particular batch of mice that one mouse completely outgrew. And so in the next, so this is essentially due to one mouse. And I can show you we have the raw data slide as well. That mouse was way up beyond where the top of the tick is. And so in terms of the statistics and the standard deviation, it makes the error bars go wonky. We could, in lots of times in science, we throw out that one because, call it an outlier. We've kept this in for full disclosure because the rest of that line is so flat and so perfect that in science, if something's too perfect, we get suspicious too. So,
and when you say perfect, these error bars suggest they're really tight results in there too.
That's perfection. That's exactly when I said earlier, this is beyond what I had hoped. When we see error bars that are that tight, that there's a complete separation that early on, it allows us the word that use synergy, which is rarely easily applied truly in science. And we use enough mice. N equals 6 is because we need enough to give us that statistical significance. Otherwise, if we used 1 or 2, then it could be phenomenal. If we use 24, it's a lot of extra resource that goes beyond proving the math and the statistical significance. And so it's just the reason. That's the reason we don't have to. But we have to repeat the experiments. So we have to see this over and over in the humanity of research. to unnecessarily sacrifice the animals. But if you do the exact same experiment over and over, then it's real. And so now we're doing it again. The same results are coming out essentially. We're varying it to have bigger.
Yeah.
There you go. Here's our slide. So
Yeah.
this is our single outlier. If you take that single outlier out, then this would uptick a little bit, but it wouldn't have caught anybody's eye because, So this is essentially, this is the raw data. So full disclosure, we're so proud of it that we want to show it off.
Here. Yeah.
Yeah. So anyway, that was essentially so that in science you just want to use you want to have the numbers big enough to allow statistical significance and then repeat them so that you've proven your.
Yeah. Question here about, in the past we've talked about colorectal cancer as being our primary focus, and that may ultimately be the big market for it. But the reason we've chosen melanoma is because we believe it's the most efficient path to effectively achieve a beachhead with the regulatory powers that be and the scientific community to show that it works in the worst of the worst cases where nothing else works. That's how you really stand out, make a name for yourself, and show that there's promise for this. And as Carmen said, you then work backwards from there. In the colon, obviously, it's complicated environment, lots going on there. The melanoma is the case we saw, a lot easier to access in the near term. So it's a question of efficiency, and we expect it to provide a faster path to first in human. Which answers a few of those questions. There's a question here about, here we go, IP. Great question. What components of the process are protected or protectable? Intellectual capital or intellectual property? How broad are the claims? Just want to see if we have that in our appendix. No, we don't have that slide. Sorry. So I will speak to the IP aspect. There was a question around the fact that it's a 2-device system. So there are a number of facets to the IP strategy here. Number one, the core is obviously the patents. We have 4 patents now, new one pending, that are at the core of it, with our gold nanorods really providing that key protection, the patent around the patent pending around that. However, that's not it, and that's not it. And you also don't want to necessarily patent everything. There's the trade secrets we have in terms of the application, the time, the duration, when to administer, dosage, etc., are all proprietary information that we're developing with each incremental study that's done. There's more and more data and experience that accrues to the company. Thirdly, there's the fact that it is a 2-device system. So between the gold nanorods and the light device, once that is, as we hope, approved and by regulators, you can't use one without the other. So if someone gets a light device, they can't do it with their own gold nanorods or somebody else's gold nanorods, and vice versa. So there's an inherent protection around that. So multifaceted protection there. We continue to develop and file for patents, and we'll continue to do that as we go forward. Other questions here?
I saw a question there, yep. David. It just flashed up, but it was about veterinarian application. Yes. And so that's another good intuitive question. So I have been in discussions with a local veterinarian about what diseases they see frequently, and one of the turns out, and I have a Bernese Mountain Dog. One of the things these animals get are melanoma, turns out. So it occurred to me, could we look at this in the veterinary world? And so we're just having early discussions now to see if there are opportunities there. I suspect there are. We're trying to develop a collaboration with one of the veterinarians to at least give us some advice on that.
Which segues us into another question here, is whether large animal studies would be required in order to progress this and obtain approval. Len, do you want to speak to that?
Yeah, so the feedback we've had from the FDA is that if we stay for the time being with intratumoral injection, local injection, not systemic injection, then we probably will not require large animal studies for safety. There may be a need for large animal studies for a usability perspective in some cases, But for the melanoma application, we suspected that won't even be necessary.
So that was a win for us because, of course, it's more efficient. It shortens the path and makes it cheaper, faster. The ability, the next studies will be done in rats. Another question here is the goal to partner off the cancer therapy commercialization, or will Sona seek to take the therapy to market itself? Great question. Is bigger goal to sell gold nanorods, and cancer therapy is effectively proving out this technology? There's great commercial questions there. So, whether to partner, it's the typical path for small biotechs such as ourselves. They're often set up to effectively prove out a technology and a therapy, and the big guys come in and in a variety of different fashions could partner, help fund, etc. What we need to stay focused on is just driving forward, proving out this concept, and it's so powerful and of interest and timely, I'd say also, that we think that those are strong possibilities. Obviously, no guarantees here, but we're excited to be able to take this to some of the scientific conferences in which these larger players inhibit. Bigger goal to sell gold nanorods, and cancer therapy is effectively proving out this tech. It's both, but I must say the bigger scale opportunity is the therapy. We really believe that. The sad fact is that while cancer rates or survivability of cancer has improved, there are many cancers for which the rates of incidence are increasing. And some of the ones that we're focusing on here. So we see that as the big opportunity in order to be able to benefit the medical community. Any other questions? Or I think I think we've covered most of it. We're always happy to take your questions. Thank you for joining today. I want to thank Dr. Jack Montonio for joining us, and we'll look forward to sharing more results with you when we have as this exciting study proceeds. Thank you, everyone.